There we go. Just as a brief legal disclaimer, all the products we're going to talk about here today are intended for molecular biology applications only and are not intended for the treatment, prevention, or diagnosis of any diseases. So our agenda for today's talk is going to be talking about what is digital PCR, and then what is the QIAcuity system that you now have on site at Yanping's lab down here at ICBR, and basically how it operates, right? So there's some unique differences from this system relative to other digital PCR systems on the market, namely droplet digital PCR, or which is sold by Bio-Rad, or the Stilla droplet PCR, which is turned to crystals, and also the Absolute Q as well, which is sold by Thermo Fisher. We'll then dive into some just analytical performance characteristics of the QIAcuity. So basically at its core, digital PCR is counting molecules, right? So we'll talk about how many of those molecules you can count. And then in the application section, we'll get into the context of this, right? So if you leave with nothing else from today's talk, I just want you to remember that digital PCR is nothing more than counting how many genomes or microRNAs or whatever nucleic acid target you want in your sample, right? That's all it is. It gives you absolute quantification of your nucleic acid targets. So, but where is it actually used? It tends to span a lot of biosectors within the biosciences. So, right, we see it in everything from cell and gene therapy applications, wherein we're titering AAV to determine how much we're actually producing from our production facility or our small scales. We see it in basic academic research across many fields. So everything from environmental DNA to molecular pathogenesis, basically everything there. We see it in oncobiology a lot, and then really in things like food safety as well. And so in terms of how it actually positions itself relative to other PCR-based technologies, I think we're all probably pretty familiar with conventional PCR, right? So we've done this in either high school or undergrad in some way or another. And in conventional PCR, what you have is your template, right? So that could be DNA or versions transcribed RNA. And what you're then going to do is put it into a tube. And in that tube, you're going to have your buffers, right, that allow for a biochemical reaction to proceed. You're going to have a polymerase, which synthesizes new strands of DNA from your template molecules. And then you're going to have things called primers, right? Those are telling the polymerase where to amplify DNA. And so after that process of thermal cycling, let's say 30, 40 cycles where you heat and cool your reaction, you in theory should get product from that reaction, right? And what we do then is we run that out on an agarose or acrylamide gel, and we either see a band or we don't see a band at the size we expect. So this is really a qualitative analysis. It's a binary. Yes, we have it present in our sample well. No, we don't have it present in our sample well, which serves this purpose for many things like genotyping or maybe a quick type of detection mechanism where you don't need a good limited detection or quantitative ability. So in the 90s, real-time qPCR came around, and this again is really the same fundamental reaction components as what you use in your conventional PCR. So you have that primers, you have your polymerase, you have your buffer, you put your starting template in. But now the major difference is within this reaction mixture, we have fluorogenic molecules. So either things like TACMAN or hydrolysis probes, which when they're cleaved are going to give you fluorescence back that you can read from your qPCR system. Or you have something like CyberGreen1 or Evergreen, which basically just bind to all double-stranded DNA as it's produced in the biochemical reaction of PCR. So ultimately what this gives us though is a rise in fluorescence over a run in time where time is expressed as a cycle, right? And you can see different amounts of starting input template will rise at different times throughout your thermal cycling process. So the earlier they rise, so the smaller that cycle number is, the more concentrated your starting material is. The later they rise, the less concentrated it is. So this is a really great technique in molecular biology for doing things like relative comparative measures. So if you want to say that I have in treatment A, a 2 or a 10-fold upregulation of my genes relative to treatment B, which is my control, qPCR is a great tool for that, right? Because you're not necessarily saying I have 10,000 more copies or 50,000 more copies of my transcript or my genome of interest. You're just saying on a relative scale, I have more compared to a control and doing the right control measurements. So where qPCR tends to fail a little bit though, is when you want to ascribe an absolute number to what was in your sample well. So there is a method for doing that in qPCR called absolute quantification. And what you're seeing here is basically a subset of those data. So whenever you want to try to do absolute quant on qPCR, you have to source a standard material that has a known identity, so known sequence. And then you have to do dilutions or standard curves and run that on every plate with which you're running unknowns. So very similar to an ELISA, right? It's an interpolary assay, because what we then do is we take the log of that starting material and we plot that with the cycle value when it rose above the background threshold. And so this gives us a linear regression. And for unknown samples, let's say we want to know where this sample lies, we interpolate onto this equation, and it will give us an approximation of how many starting copies we have. So qPCR is great for many things, but it's one of these techniques where it's really easy to do, and it's really easy to generate data. And anytime you have an analytical technique like that, it's really easy to get into trouble, because you have to make sure that you're staying within the boundaries of things like the qPCR MICE guidelines to make sure that your data are actually analytically accurate. So a number of things which affect qPCR's accuracy and analytical capability are the reaction efficiency. So how close are you to a perfect doubling of that template with each subsequent cycle? Things like how well does your standard curve actually match the background of your unknowns? And the further away you deviate from kind of that perfect scenario, the less analytically accurate you are, and you just have this unbounded unknown error, right? So it can really start to bloat whenever you're doing absolute quantification. So digital PCR is very similar to qPCR in what you actually have in the reaction. So it still has your primers, your probes, or your double-strand binding dye, your polymerase. But now what we're doing is, instead of taking that reaction and putting it into a single well of a 96-well plate or a single tube, we're taking it and we're fractionating it out into many, many thousands of different little micro reactors. So it's a microfluidic system, all right? And there's different ways you can create these microreactors. So some companies do it with emulsions. So basically water and oil droplets. Other companies do things very similar, but they call them crystals. The way QIAGEN does it is we manufacture these partitions into our plates that you're going to use on the dPCR system. But the take home here is really that now we have an independent and random segregation of our template molecules and our reaction mixture into, let's say, 8,500 or 26,000 partitions, right? Within each of those little microreactors, then, we'll either have template or we'll not have template. We could have 1, 2, 3, 4 n copies of template, but there will be template there. So at the end of your standard thermal cycling protocol that you would use for qPCR, you're going to see partitions that have fluorescence and partitions that don't have fluorescence. So from that, it actually fits a distribution known as a Poisson distribution quite nicely, where we can then just do a little bit of math and calculate how many copies per microliter of your targets were present in that sample at the start. So, and this is true absolute quantification. Any questions about that before I go on? Yes, so we don't use emulsions or droplets. What we use is the plates are actually manufactured to have all these partitions in the well. So they're essentially stamped into the plate at the manufacturing facility. And Danielle to the rescue. Thank you very much, Danielle. So this is one of our nanoplates that I'm going to talk to you a little bit more about. We can pass this around, but essentially you'll see it has a standard SBS format. Thank you. It has a standard SBS format, so like any other 96 or 384 well plate. And if you look on the bottom of it, you'll see the partitions as you hold it up to the light. So they come from the factory like that, essentially. So you don't have to do any complicated emulsions or processing to the actual plate itself to get those. So some of the benefits of doing digital PCR are that you have this absolute quantification without the need for a standard curve. This is really helpful for things like viral titer or counting microbial genomes, because you don't have to run that standard curve, source it, worry about stability in the freezer, and so on. We have a very high tolerance to inhibitors for a reason I'll show you a little bit later. But the take home to that is we don't care when one of these partitions became positive, just that it is positive at the end of our thermal cycling. In real-time qPCR, you care very much about when something became positive. So inhibitors tend to affect digital PCR much less than they do in real-time qPCR. The precision for digital PCR, because we're subsampling that same reaction many thousands of times over, tends to be excellent, on the order of 10% or greater. So that allows you to resolve very small fold change differences for things like microRNA or mRNA expression. So the increased analytical sensitivity allows you to detect very rare targets. So think about SARS-CoV-2 in wastewater that maybe you've read about in the news, right? So this system, digital PCR in particular, has been adopted by about 70% of the US states for their public health monitoring of wastewater systems for SARS-CoV-2. And then very high reproducibility, so on the order of inter or intra-operator, intra assay, and then across site studies. So it makes it great for these longitudinal studies that involve many different institutions. And here's just a summary of really what we just talked about. So qPCR relies on standard curves for absolute quant, has a high reliance on amplification efficiency. It's very sensitive to inhibitors in that they're going to push your trace back whenever you do run it. But you do have real-time detection, which is helpful for library prep sometimes, and you can get melt curve analysis, which is also a benefit for certain situations. So digital PCR has no standard curves required, relaxed efficiency requirements, high precision reproducibility, that less sensitive to inhibitors, but it is endpoint detection, right? So we're not having that real-time detection for things like library prep. And here's just really an example of that 10% precision in play. So usually whenever we're thinking about what qualifies a difference, I know I'm talking about qPCR a lot, but it's a thing that most people are familiar with. So I tend to kind of frame it in this context. Whenever we're thinking about what's the significant difference in qPCR, generally it's a one cycle difference, right? That's going to relate to about a twofold change or 50% change. So within one cycle though, we can do about 8 dilution points and resolve those cleanly from one another and basically have that enhanced resolution within essentially what you would have seen in one cycle difference of qPCR. And in terms of the actual processing, it's exactly the same as what you would do in qPCR. So you mix together your template molecule, either any type of nucleic acid. You can start with RNA, DNA, cDNA, whatever you like there. We're going to mix that with our reaction mixture, which is basically a master mix you buy from QIAGEN. We're going to pipette that into the nanowells of that plate that's going around. We're going to apply a rubber seal that's also going around. So this is basically like a plasticized rubber that goes on top of it. And then we're going to put it into the instrument and walk away after we execute the program. So it's going to automatically do the partitioning. It's going to do the thermal cycling and then the data acquisition by itself without any other intervention from you. And just in terms of the relative workflow, so if we think about real-time qPCR, you're looking about 1.5 hours to 2 hours for standard cycling conditions for your time to results. Bio-Rad's droplet digital PCR system is a little bit more intensive in terms of how much hands-on time you have to do, because here you're loading cartridges with your reaction mixture, generating droplets, transferring those droplets, sealing the plate on an off-board thermal cycler, then doing the readout on this modified flow cytometer. So it takes about 5 to 6 hours, all things told, until you actually have a data file to analyze. With our nanoplate that you see here, and because we're not doing any of that droplet generation or singulating those droplets through that flow cytometer-like system, you're going to get to about under 2 hours or around 2 hours for your data analysis, depending upon your thermal cycling protocol. So it's very analogous to qPCR in your time to answer. So we have 3 systems, and the one in Yanping's lab is the QIAcuity 1. So with this system per 8-hour shift, so basically let me back up. So this can do 1 plate at a time. So you basically take your samples. I'll let Yanping speak at the end to talk about practically how we do this. But you take your samples, you mix them together, you pipette them into the nanoplate, you put it onto the QIAcuity and execute the program. So it processes 1 plate at a time. And you can have about up to 384 samples per 8-hour shift if you're using one of our 96-well format plates that you see coming around, or 96 samples if you're using one of our 24-well format plates that you see coming around as well. So your time to results is 2 hours, and I believe they have that in the 5-plex configuration. So you can do 5 independent targets on a per well basis because we have 5 fluorescent channels. And the lab footprint's actually quite small. It looks a little bit bigger than old. I'm not going to date myself about how big computers used to be, but it looks like an older computer. It's a little bit larger than that. So that's the system itself. The actual consumables you use, one of those is coming around, are going to be your nanoplates, your reaction mixtures, and then your assay. And that's all you need for this outside of your sample. So we have 2 different nanoplate densities. You see your 26K 24-well nanoplate. This has 26,000 partitions per well and allows for up to 28 microliters of sample. So this is for rare event detection whenever you don't expect to have a large amount of whatever it is you're looking for. So this could be trace microbes, it could be liquid biopsy research studies, basically anything where it's going to be rare in your sample type. And this affords you the resolution in that large sample input volume to get the detection sensitivity that you need. So the other plate that is actually in your hands here today is the 8.5K plate. So this has 8,500 partitions on it per well. And so this is great for applications where you have to dilute your sample down to get into the dynamic range of digital PCR. So things like copy number variation when you're doing cell line engineering and development, things like viral titer, any type of microbial titer, or for routine gene expression or microRNA, any type of RNA analysis. It tends to work well for that. So in terms of our actual chemistries, we have a hydrolysis probe kit. So for any assays you're using now on qPCR or droplet digital PCR, we have a very high success rate of porting those assays over, just modifying a couple of things like annealing temperature or maybe the probe's and primer's final molarity in that reaction. So we also have an evergreen kit if you want to use primers only. So let's say you're doing cybergreen today detection, and maybe that's not giving you the resolution or quantification you need. You can transfer it over to digital PCR. And then if you have RNA targets or RNA plus DNA targets that you want to co-detect in the same reaction, we actually have a one-step advanced kit that allows you to put in just your RNA. It'll go through that RT to make cDNA, and then the amplification in the well. All right, so here's our plates. We talked a little bit about it, but then actually if we dig into this a little bit more, for any one of these wells, what we're going to see is 4 unique features within it. So there's an input well where you pipette, or you have one of our liquid handling robots pipette into it. There's a vent port because we're going to pressurize the liquid that goes into here. And then you see the microfluidic circuitry that kind of goes in between that input and the vent. And this is basically where your reaction mixture is going to live for the thermal cycling and detection. Right? So how the system actually operates, I'm going to use a video to show you that, because I guess a video is worth more than 1,000 of my words. So when you first put the plate in, it's going to scan the barcode that's on the plate for traceability purposes. And then a robotic arm is going to grab that plate and it's going to move it over to the priming module. So this priming module is where your partitioning occurs. And you remember that seal that I told you about? The way this essentially happens is the plate's pushed in, and then there's this little piston headset that's going to come down and actually physically contact that rubberized plastic seal. And that's going to provide the pressure which is needed to force all of your reaction mixture through the connecting channels into the individual partitions. So once that's been completed, a roller will come on the underside and then hermetically seal off those partitions such that they're discrete individual units and won't merge during thermal cycling or additional handling or data acquisition. So the robotic arm comes back in after that routine's done, and it's now going to take your plate to the next area of the instrument operation, which is going to be the thermal cycling module. So this is, you can think of as a standard flatbed thermal cycling. It's going to do your hot start, it's going to do your denaturation, and then your combined annealing plus extension phase, similar to what you would on qPCR. And so in this step is where we either have amplification or we do not have amplification of our target molecules across those 5 channels. All right. So how we actually detect whether or not we had amplification or no amplification is by moving it then to the next module, which is the data acquisition module. So this is basically just going to image the plate. So what we're going to see here is on a partition by partition basis, well by well basis, we're going to scan with a bright white LED and the corresponding channels that you select. We're going to collect the resultant fluorescence data down through a camera and actually get a physical image of that fluorescence for every partition in the actual plate. So what happens with that then is we move into the software for analysis as end users, and we'll get our copies per mic out. So what we as end users see on a well per well basis is going to look very similar to this. So on your y-axis here, you have your fluorescence intensity expressed as RFU. On your x-axis here, that's just a running plot of your analyzed partitions, right, as they've been scanned by that imaging system. So what we see is we're going to basically set a threshold, or we'll allow the software to set a threshold, and then that delineates the positive high RFU population from the negative low RFU population. And then that proportion of positives to negatives is what goes into the Poisson calculations to give you copies per microliter. So if you're looking at more than one dye at a time, we have 2D plots. So basically you would select which channel you want to see on your y-axis, which channel you want to see on your x-axis, and it's going to plot out a similar fluorescent, basically scatter plot here. So if you're low on your y and low on your x, these are dual negative for your targets. If you're high on y, low on x, this is your green only positive. Let's say high on x, low on y, this would be your yellow channel only positives. If you're high on both channels, this is where you had co-partitioning of 2 independent molecules into the same partition, and they both amplified. Any questions before I move on? So are you saying, can you do more than one target per well? Well, so your number of master mixes depends on how many targets you want to detect per well. So if you want to do, let's say, 3 targets per well, you would combine your super mix or your dPCR mix with that, and then your corresponding primers plus probes into one master mix. And then you would pipette that across your wells and then add your different samples. If you were doing 5 targets, let's say, it would be the same idea, but you're just adding in those additional 2 target primers and probes. Yes, so you could do different primer sets per well, or you could do all the same primer sets per well. It just depends on what your experimental design would be. So like what you want to detect actually. Yes, so that was, so in the video it was showing one individual well, but basically, yeah, so depending upon what you want to amplify on a per well basis, you'll do the correct master mix for that individual well. Because as you saw on the plate, it has wells just like any other 96 well plate. Right, so if we were doing, let's say, oh yeah, I'm sorry. So thank you very much. Yes, so basically, I mean, this is your standard 96 well plate, right? So it has A through H, 1 through 12. So if I wanted to do, let's say, target, I don't know, gap DH in this well, and let's say HPRT in this well, I would make up one master mix for gap DH, pipetted across this column, one for HPRT, pipetted across this column, and then you get the corresponding detection wherever you put your master mix. Yeah. So actually, if you look down below here and kind of hold it up to the light, you can see the little squares correspond to the per well partitions. For 5-color, I don't have one here today, but I can pull it up probably at the end to show everyone. Yeah. Yeah, so for 5-color analysis, essentially we don't have any 5D plots yet just because our brains tend not to work well in 5 dimensions. But you just iteratively choose what you want to see on your Y and X axes to do that in 2 dimensions. So your software is going to look very similar to any existing dPCR software that you may have used in the past, in that we're going to have our 1 or 2D scatter plots. You'll have your secondary analysis tools like concentration or point diagrams. One thing that is different is you're going to actually have these raw data to go back to. If anything may look skewed or off, you can always do some quality control on those plate images. So in terms of the QIAcuity system performance, one of the important things about digital PCR is to have an expected number of partitions that you get day after day, assay after assay. And so QIAGEN went to great lengths to actually do quality control on these plates to ensure that you're getting about 95% recovery of your usable partitions run after run after run. And so for other systems, you'll notice someday you might get 10,000 partitions, 16,000, 20,000, then back down to 12,000. And that kind of creates this unbounded uncertainty associated with your dPCR experiments. Here, the real take home from this is over 15 runs for 8.5K and 26K plate, we're seeing an expected number of recoverable partitions. Then in terms of the actual dynamic range of the system, so for that 8.5K plate, on the lower limit, you're looking about 17 copies per reaction. On the upper limit, you're looking about 170,000 copies per reaction. So that's a per well basis, that's your dynamic range. For that 26K plate, you'll remember I mentioned it's for those more rare targets. You're looking about 6 copies on the lower limit and about 220,000 copies on the upper limit. And so that's basically for the rare event detection, varying a little frequency if you're doing, let's say, oncogenomic research and so on. And if you've used dPCR in the past, you may be wondering how the system actually performs to other systems on the market. So here we have a comparison for 3 different genomic targets, CTA-NNB1, EGFR, and ERBB2, across both our 26K plates, our 8.5K plates, and then the BI-RAD QX200 at 2 different dilution points. And so you can see comparability at 100 copies per mic input and 10 copies per mic input across here, right? So it's basically, you'll get analogous data coming out because it's still digital PCR. We're just doing it with nanoplates as opposed to droplets and faster. So for our example applications data, if you need any assays, so you can either design them yourself, you can use your existing assays that you have for your targets, or you can buy them from QIAGEN. So we have our GeneGlobe website, which is actually a great tool that helps you explore, plan, analyze, and design your experiments. But we have basically assays for mutation detection. So if you're looking for SNPs relative to wild type, for copy number variation, for microbial assays, and then also for microRNA and mRNA expression types of experiments. So it's a great resource for your experimental needs. But first, we'll talk about mutation detection. So really, if we think about the designs, they're pretty much the same as you would use for qPCR, right? And a lot of times, whenever we're doing SNP detection or any type of, let's say, indels, what we have is we want to detect our mutant sequence and our wild type sequence. We want to know the relative abundance thereof. So we'll have a common forward and reverse primer, which target both of those flanking ends of our mutant and our wild type. And then we're going to have these discriminatory probes. So one colored for the mutant, one colored for the wild type. And then we have 5 channels on the QIAcuity, so you can select across any of these dyes from green, yellow, orange, red, or crimson, and then match them with very common commercially available dyes that are present. Things like FAM, Hex, Tamara, and so on. So what we ultimately see whenever we're doing these snippet types of experiments is going to be shown by this example here. So this is using one of our 26,000 K plates. And here we basically just spiked in Horizon Discovery FFPE, fixed formalin paraffin embedded tissue samples. So these tend to be very difficult samples to work with. They've gone through that fixation process and that paraffin embedding process, which creates things like inhibitors, abasic sites, and really tends to, I guess, muss up your downstream applications. So we spiked 0.3 nanograms of that into 30 nanograms of universal human reference DNA, and we measured the mutation frequency with one of our dPCR SNP assays. And what you can see here is an example 2D plot. So here's your negative partitions. They're low for both. On your y-axis, this is for a target called BRAF V600E, which is known to be implicated in a number of different cancers. We see this is our wild type population here. On your x-axis, this is your V600E mutant. And then we have this one lonely partition up here where you had co-partitioning of one wild type and maybe one wild type and maybe one mutant into one partition, but it's going to show up as one. So in total, we have 6 copies per well, which gives us a 0.1 mutation frequency, right? So if you wanted lower than that, you can always, let's say, because the wild type is really what determines your variant allele frequency. If you want to push it down and detect more rare targets, you can pipette across multiple wells and then merge those together in the analysis. So copy number variation tends to be another popular application for digital PCR in that it's basically just more amenable to digital PCR and things like qPCR. And here we're talking about genomic structural changes, right? So do you have things like aneuploidy, where you have an increase in number of chromosomes? Do you have segmental aneuploidy, where you have things like duplications or deletions of certain chromosomal regions? Or are you doing things like cell engineering, wherein you want to confirm that you're putting the right construct into your cell line whenever you've done something like AAV transfection or whatnot? So these assays tend to perform really nicely. They're 2 independent targets. And this is an example from one of our customers at Gothenburg University in Sweden, who was basically trying to understand the correlation between amylase 1A. So it's an enzyme we all have in our saliva and our mouths, I should say. And basically, it's known to vary in copy number state. So there's a huge range of copy numbers for AMY1A. And that's the variable target. So here is a stable reference, AP3B1. So that's known diploid single copy across humans, right? It tends to be, I believe, lethal to the cell if it's duplicated, inverted, deleted, things of that nature. So this researcher took donors that had this orthogonally validated by NGS and FISH samples with either 4, 6, 8, or 17 somatic copies of AMY1A. And they wanted to measure to see how well digital PCR could resolve those copy states. And this is just basically a representative distribution of those experiments. So for donor number 4, what did we get? We got a copy of 4. For donor 6, we got a copy of 6. For these 2 donors that had 8, we're getting 8. For those 2 donors that had 17, we're getting 17. Then over here, you can see the copies per genome and the relative confidence interval associated with that. So this is why digital PCR tends to be a really great tool for copy number variation is provided you don't have heterogeneous samples, you're going to get these on integer calls. Even if you do have heterogeneous samples, things like fine needle aspiration biopsies, where you have maybe, let's say, 10% of your cells are cancer, but 90% are wild type, you can still detect copy number changes and correlate that back to your actual pathology. Any questions before I dive into pathogens? Anybody here work on pathogens or microbes? Okay, thank you for the little hand. I appreciate it. All right, so we're actually really excited because, for the past 2 years, we've been hearing about SARS-CoV-2, right? Yes, we do have SARS-CoV-2 assays. We have a ton of them, and we do it for wastewater. But we just recently launched a assay offering of about 685 different microbial pathogenic and then just microbiota assays for the QIAcuity system. These are all probe-based assays with about 200 of them being bench validated in terms of their performance and flexibility together. And so you can choose any one of our 5 dyes to mix and match for these detection panels. And basically, you would need this assay, you would need your sample, and then you use one of our 2 mixes. So if you want to do, let's say, RNA-based viruses with DNA-based microbial detection, you can do that in one reaction with our 1-step kit. And so a lot of these assays, at least for the microbial or the bacterial targets, have been designed based upon 16S ribosomal RNA similarity. So we all know that in 16S ribosomal RNA for bacteria, you have conserved regions and variable regions, which match back to your genus, species, and then strain identity. And so we basically focused in on these variable regions to seat our primers and probes. And so you heard me mention these 5-plex wet lab tested bundles. So this could range from things like cannabis production, virulence for AMR, resistance genes, and everything like that. Also, we have wastewater samples as well. So if you have any things that you would like to see as you move forward for either environmental monitoring, wastewater monitoring, human microbiota, or non-human microbiota, please do let us know. And we're certainly open to at least having some testing of those panels in our Germantown Labs lab. You may be wondering how it performs, and I do have a slide for that. So here we're using our one-step advanced probes kit. So this is where you can put in either DNA or RNA. And so we did a 5-plex reaction here for Vibrio, Legionella, Yersinia, Listeria, and SARS-CoV-2. And we did this at two different dilution points, 500 copies per mic input, 50 copies per mic input, and we had a no template control along with that. And so you can just look across the top here at your 500. You can see that we're very close to that value. And down here at your 50 copies per mic input, you can see we're very close to that value as well. So it performs really well in this co-detection type of environment with nice, clean, no template controls. So for RNA expression, which I believe is my last application here, let's say you have something in your qPCR reactions where you're basically beyond cycle 30. And this is going to differ for everyone if you're looking at low-level expression. Usually beyond cycle 30 or 32, you start getting your standard deviation starting to bloat. Oftentimes after cycle 35 is where qPCR reactions really break down. And that's due to a number of reasons we won't have to get in here to today. But essentially, that creates this kind of gray zone of what do you call that, right? Because you have this huge standard deviation associated with your low copy reactions. And so this is where digital tends to be a really good, almost reflexive technique where you can test on qPCR for your experiments, and you can move to digital PCR if you need more resolution for those low input samples, or you can just port over to digital PCR for your routine experimentation. So the reason why this tends to work well is shown here. So basically we have two different targets, IL-4. We have three targets, sorry. We have two reference genes. IL-4 is our target, HPRT and GAPDH are our reference genes to which we're normalizing. And this is a synthetic spiking experiment wherein we have 4 different samples. So sample 1 should have an expectation of 0.17-fold downregulation. Sample 2 is our reference. Sample 3 would be 0.17-fold upregulation, and sample 3 would be 0.67-fold upregulation. So for those of you who work with fold regulations, you know, that's very good resolution, right? Generally, you're looking at like a 2-fold change, maybe a 1.2-fold change for neurobiology applications. But what we can see here is, according to these spike-ins, we had our expected values, so 0.83 down, 1.17 up, 1.67 up, and then our actual measured empirical values. So 0.85 down, 1 will always be 1, that's a reference, 0.23-fold up, and 0.67-fold up. So if you are working with these very low expressors or you need to resolve small fold change differences, digital tends to be a great technique to go with. Also, if you're working with difficult sample types, things like, let's say, microRNAs in different biofluids or different materials. So pretty much here what you're seeing is copies per microliter on total RNA input across 4 different microRNA species, right? And so you have some things which have very high amounts of microRNA, which are pretty easy to work with, like your cell lines, your Buffy coats of your extracts, and so on. Then we're kind of going down the list here in terms of amount and kind of difficulty to work with. So you have your FFPE, then you're going down to actually extracellular exosomal DNA, your sera, your plasma, and then very difficult, nasty inhibitor-rich things like urine and cerebral spinal fluid, which you can't either get enough of for CSF or for urine, which tends to have just a ton of inhibitors present. So what we can see is down to about a picogram of input, you know, high accuracy copy number detection for microRNAs. And then this is just really a comparison of dPCR relative to qPCR. We know there's a lot of microRNA work done with qPCR. So if you're looking at 10 copies per mic input, your blue bar is your dPCR here. So your red line is your qPCR. And you can see they're pretty good, matching over to CQs of about 30, maybe 31. But then once you get down in a 10-fold dilution, you go to about 10 copies, what you can see is dPCR is going to give you that reliable, accurate copy count, and qPCR is going to bounce around with high CQ values. All right, so in summary for today, just like I said, the QIAcuity system presents a simplified and rapid approach for digital PCR applications, and it provides you this benefits like absolute target quant, high tolerance to inhibitors, 10% precision, increased analytical sensitivity, and high reproducibility. With that, I would like to thank you very much for your attention and take your questions that you have. So we have about 685 total bacteria, viral, fungal assays which are in our portfolio, but 200 of those that I mentioned are wet lab tested, where they're tested for flexibility and just overall performance. But of our actual microbiome assays, I don't remember the exact number we would have, which would be relevant, because everybody's looking for a little bit different targets within that. So we do have a great, 20-page, I think, brochure, which lists out all of our assays. And so we can usually send that on to search, and we can send it on today.